the session later on. All right. So in the previous the lecture I taken on I think on 30th of April. So on 30th of April I discussed about the databases. So what are databases? The building blocks of the databases, the benefit of the databases, the characteristics of the databases, the DBMS database management system. That each and everything I discussed in very detail. So now you do have the idea what database is all about. Concurrently, thousands, lakhs of people can work on the database. Just like I gave the example of the Facebook. Millions of people are using Facebook at the same time. So that is nothing, that is the miracle of database only, right? So if we can put our normal information on the database and people can access throughout the world, then why we cannot put the biological information in the database? Whatever data is generated in our laboratory, if that we keep posting on the database, so the researchers who are located around the globe, throughout the world, they can access that biological data and they can think beyond it. Say for example, if some country have got some variant of the coronavirus, so if I do the sequencing of that particular coronavirus for that particular mutant and I can make it available to the database, I can submit to the database. So definitely that database, that sequence will be available in the database and anyone sitting over any part of the world, he or she can access it and they can carry out the research on that, right? So this is the benefit of the database. Within a fraction of seconds, your data may be reached to the public, may be reached to the researcher who want to pursue the research on that particular sequence. So biological databases, simply data in databases, if we put the biological data, that is simply known as biological database. Meanwhile, if you have any question, unique curiosity if you have, you can keep posting on the live chat. I will be very happy to answer you. If you have any question, you please ask. So, I talked about the normal characteristics of the databases. Now today in today's session, we are going to discuss, we are going to talk the type of the databases and what are the different major databases available in the public domain in bioinformatics that you have to study and that is beneficial for the microbiologist or any life science researcher. So these are the contents that I am going to cover today, the features of the biological database, the characteristics of the biological databases, and obviously the classification of the biological databases, that how many type of biological databases are there. So first is biological database. Why we need the biological database, right? So why? Number one, need for storing and communicating large data set has grown. If you are aware about the sequencing, once I was talking about the sequencing, I was talking about the history of bioinformatics in that I told you. First time the protein sequencing was done by Frederick Sanger and that was of insulin in 1953. So the first sequencing technique was developed the protein sequencing method, then it was the RNA, then DNA, right? So for DNA, lot of method, lot of generations are there. Now third generation of sequencing is going on and this is known as NGS, next generation sequencing. So if I give you an idea, the crude idea so that you can understand, if you know about the Human Genome Project, so the Human Genome Project was started in the late 1989, so officially you can say it is in 1990 and it was declared as an end in or completed in 2003. So it was taken 13 years of effort, thousands of scientists worked 24/7. Then scientists were succeed to sequence the single genome of a human, right? So that was a 13 years effort and you will be surprised to know that whatever work was done in 13 years of research, rigorous research, now if you want to do genome sequencing today, that can be done in a day only. So 13 years of work is equivalent to 1 day work now. So our technology is growing very faster. And once I was talking about in the history of bioinformatics, in that I told you that due to ethical concern, none of the single experiment was done on the human genome, on the human. Then how we annotate what is the function of any particular gene, what are the different genes, right? So definitely the work was done on organisms, similar organisms, those who have the similar genome. So you can carry out the work on the organism, the model organism, and whatever result you get, that you can impose over the human genome, right? So day by day, researchers are doing the sequencing of the different organisms. So what happened? Now this is the flood of the biological data, and that massive amount of the data, if we want to manage, if we want to spread throughout the world, then we have only one option, that is the database. If we use the database, we can spread our biological information to the scientific community. All right. So obviously, the point number one, the need for storing and communicating large data set has grown, which is grown at a very massive amount. Number two, obviously, we want to make biological data available to the scientists so that they can think beyond that whatever have done till date, they can think beyond it. Number three, to make biological data available in computer readable form. Obviously, if we make the biological data in such a form so that software can read it, because with the help of pen and paper, to analyze the biological data is near about impossible task. So there is a need of some common format so that the software can read that particular biological data and we can analyze that, and something which is novel, we can figure it out. So what are the features of the biological databases? These are the different features: the heterogeneity, the high volume data, the data curation, the data integration, the data sharing, and the dynamics. So let's see one by one the heterogeneity. What data heterogeneity means all about. So in the biological data, as biological experiments are concerned, there is a lot of heterogeneity. There is a lot of different type of the experiments that we conduct in our laboratory. So there is the heterogeneity in the database. In the biological data, there is the heterogeneity. Like the sequence may be the biological data may be of sequence. So maybe it is nucleotide sequence, maybe it is protein sequence, maybe it is graph. What graph is all about? Graph means the data indicating relationship among themselves can be captured as graph. Say for example, the pathway data. I am sure in biochemistry you have read about the glycolysis, the uh, different type of cycles in plants, that is C3, C4, the photosynthesis, right? So all the pathway you might have studied, and that can be uploaded on the database with the genetic map, the structural taxonomy. So these are the biological data which can be represented, it can be submitted to the database in the form of the graph. The high dimensional data, the data generated from microarray. Microarray, I think you know about the microarray. Microarray means it's a single chip. In that, if you are using that particular single chip, in a single chip, you can get the answer of two questions. Number one, in a particular condition, in a particular situation, how many genes are expressing, right? How many genes are expressing at that particular moment? And if gene is expressing, means it is on. If any particular gene is not expressing, means that gene is off. And if it is expressing, then what is the level of expression? Level of expression means, let me give you one very simple example. If a newly born baby comes in your family, right, and you go somewhere, after 1 month you come, you will see the noticeable difference in that kid. Noticeable difference in terms of his growth, the height, the weight. But if you see a teenager, a 16-year-old, after 1 month, you will not be able to see the noticeable difference. Although the growth is happening in both the cases. In kid, it is going on, and in the teenager, it is also going on. So growth hormone is releasing in both the cases. but the level of expression per minute, per second, or per hour, or per day, how many number of mRNAs being formed. So that shows the expression level. So you get the two answers in a particular condition. What are the genes which are expressing, and if any particular gene is expressing, what is the level of expression? You can quantify the level of expression. So once you conduct the microarray type of experiments, there is a huge amount of data generated, and that is known as high dimensional data. So these all comes under the data heterogeneity. The shape, and the biological data is shape. Shape means 3D structure. 3D structure of the, say suppose you have any particular protein. So for that particular protein, if scientists want to study the 3D structure of that particular protein, there is three methods. And these three methods are like X-ray crystallography, the NMR, the electron microscopy. So these are the three methods. With the help of that, you can determine the 3D structure of any particular protein, so that you can study in detail about the protein. So if any researcher is doing the structure determination, that comes under the shape, and that can submitted to the database. So that is the different type of the database. Third, next, temporal data. Temporal data means for studying dynamics of any biological system. So for example, in the developmental biology, pattern. So there are pattern lying within the genome that characterize biological entities like regulatory sequence, like promoter, operator, enhancer, silencer. So these are coming under the pattern. Extracted feature data means numerical data obtained from combination of one of the above mentioned data types, that comes under the extracted feature data, right? So these are the different that I told you about, the data heterogeneity of in the biological data. The second is known as the high volume data. In high volume data, I told you, if you are doing the experiment like microarray or SAGE or EST clustering, in a single experiment, the huge amount of data is generated. So to handle the biological data is not an easy task, right? So as our technology is going on, within less, by spending less money, we are generating more data. So day by day, our technology is growing, and more number of biological data we are generating. So this is the new challenge before the scientist, how we can cope up, how we can generate how we can develop the technology. So whatever biological data is coming now in the scientific fraternity, that we can make it available to the world. So high volume data is another feature, characteristics. The data curation. So since the biological data are collected from various sources across different structural and functional boundaries, means if you are reading in the B.Sc Microbiology, you are the B.Sc Microbiology students, and if everything is goes well, and suppose in your college you are being allotted as a dissertation, if you do any dissertation and if any fragment of DNA, you do the sequencing, you also can contribute in the database, you also may be the part of the database, you also can submit the biological data. Just like if you go somewhere, roam somewhere, click some nice pictures and you upload on the Facebook, you upload on the Instagram or any social media sites, means indirectly or directly you are contributing in the data. You are giving your data to the social media sites. So the amount of the data that Facebook have, that Instagram have, that is increasing day by day. Likewise, if you conduct any kind of the biological experiment, and that data you can make it available to the scientific community by using biological databases. So since the people throughout the world, researchers throughout the world, they are working with the different platform in the different organization, working with the different laboratories. So there is the data heterogeneity. So since the biological data are collected from various sources across different structural and functional boundaries, so there are always chance of missing links. Some missing links may be there, and that missing links can be filled by analyzing the data and by curation via automated methods. So data curation is another characteristics of the biological database. If you need any kind of the clarification on any particular point, whatever I am discussing right now, please let me know. I will explain it again. So the data curation, the data integration. What data integration is all about? So after years of research across different structural and functional scale, data is collected from laboratories worldwide and integrated together through a database and made available for use. Let me explain it, let me elaborate it so that you can understand what data integration is all about. Data integration is all about, now in coming in couple of minutes I will explain you the classification of the biological database. In that I will tell you that there are the different databases for the nucleotide, for the protein sequence there is a different database, for the protein structure there is database, for the literature means for the research paper there is the different database, alright. So suppose, say for example, if you found any kind of the organism, say for example a new virus or a new bacteria. So for new bacteria, once you do the DNA sequencing of that particular bacteria, definitely you will do the genome sequencing and you will make it available to the nucleotide database. And obviously, once you have done the nucleotide sequencing of that particular bacteria, that bacteria is also producing some protein, right? So if some researcher have done the protein sequencing, when thousands of proteins are there, out of thousands, suppose one or two or 100 or thousands of protein sequencing have been done. So for the same bacteria, there is one database in that you will submit the nucleotide, and in another database, which is protein sequence database, you will submit the protein sequence related information. And if you have the structure of any particular protein, you need some third database, that is the protein structure database. So suppose you are the researcher, you are working on some particular bacteria, so you have to retrieve the data from three databases. What? First you have to go on the nucleotide database, then protein, then protein sequence and protein structure. So what data integration is all about? Suppose I have done the sequencing of one bacteria and I have submitted in the one database, that is the nucleotide database. Now obviously the protein related information will be in another database, sequence related and structure related information will be in another database. So what we can do in the nucleotide, if concerned protein is there, we can give the link, so that if any researcher is searching for that particular bacteria and they found the gene sequence, and if they want to know about the protein, okay, excuse me, just a minute. So sorry for the interruption. So we'll continue. So I was talking about the data integration. Data integration means, I told you, If you have any kind of the nucleotide sequence, suppose you are searching for any particular gene, so once you do the you surf the nucleotide database and you found that particular gene related information, and if you want to know the concerned protein of that particular gene, then you don't have to go in the protein database and do the query from starting from the scratch. No, not at all. In the same page you will get the link for that particular protein. Once we'll go for the practical portion, I will let you know, I will give you the practical session over it, then it will be more clear. So in one database, if you get the information of another database, if you get the link of another database, that is simply known as data integration. Let me clarify it more. Suppose if you are searching for anything on the internet, right? So there is the one concept that is known as hyperlink. So and hyperlink, the feature of the hyperlink is simply that is blue and it is underlined. If you click on that, another page will be open. So just like that, if you are searching in the nucleotide database and you get the link for the protein database, so that is simply known as data integration. Means in one database, if you get the link of another database, that is known as simply data integration, right? So this is another, in all the databases you will find such type of features. Another is known as data sharing. Biological data is shared by database. What is the purpose? So that for scientific community can inspect it, cross verify it, and obviously if you are making it available one database, one data, so if you are making it available to the database, the repetition will not be allowed, right? Just like your Aadhaar card number. If someone got vaccinated, and obviously for the vaccination Aadhaar card is necessary, so once they put the person, they do the they entry the Aadhaar card number, and if person got vaccinated twice and third time if he go somewhere for the vaccination, it will be just well known that once the employee over there, the doctors or nurses or staff, once they will put the Aadhaar card number, it will show that that person have been vaccinated. So repetition like that, if I submit one particular sequence for any particular organism in database, if again someone want to submit it and that is the same sequence, that will not be allowed. So repetition, you can stop, you can prevent the repetition, and obviously you can validate the data. The dynamics. Dynamics means what? What the databases are dynamic in nature. Dynamic in nature means what? Suppose in the Facebook. So in your Facebook, your account is there, right? So once you go somewhere and you click on some picture and you can put it over the database. So what? Your information have been now changed. You changed your status. You changed your profile picture. So that is of what? That is dynamic in nature. It is not like that also made your account in the Facebook or in any other database. It will be like same throughout the life. Not like that. You can make the changes. Likewise, in the biological databases, day by day, in Word, research is going on. So biological data is growing day by day. So it is dynamic in nature. It is keeping changing. It keeps changing, right? Dynamics. So new data is generated every day in laboratories, and sometimes this new data contradicts with the old data. Sometimes it is also possible that some researchers have done something and you also do the same research and you contradict. Contradict means suppose someone saying that this is the gene, the sequence of this gene is this one, and you contradict. So that is also possible. Contradiction is also possible. So it is necessary to develop new organizational database scheme to incorporate new data. So what are the characteristics? So whatever in the 6 points I told you, that are the feature of the biological data. Now we are going to talk about what are the characteristics, what are the features of the biological database. What are the features? The number one feature is heterogeneity, that we talked about, the features we talked about. Now we are going to talk about the characteristics of the biological database. What are the characters? What are the characteristics? The number one characteristics of the biological databases are they are autonomous. Autonomous means what? Everybody is free to make the database, right? Autonomous. Autonomous means suppose I'm the person of bioinformatics and some idea comes in my brain. So I can make my database. Anyone can contribute in the database. Or I can develop database from my side only. Only whole and soul I am responsible for that. So likewise, throughout the world, different countries are there, different laboratories are there, different research groups are there. So anyone can make the database. That's why it is being called as a autonomous. All the databases are autonomous. They are made by different research groups, different organizations, different countries. So all the databases are autonomous. There is no central control over it. So the point of historical fact, most biological databases have been developed and maintained by individual research group or research institutions. So initially, these databases were developed for individual use by these group or institutions, and even when they proved to have value to the larger community. Data management practices peculiar to those groups remained. What does that mean? Means if I am developing the database, if I made the biological databases available, so I am free. I can opt it. I can adopt any technology, right? There is no control over it. It's my database. I can adopt any particular technology, right? With the data management, with the and it depends on my need, which type of database I am developing, what type of features I want to integrate in particular database. So as a result, biological databases almost always have their own governing body and infrastructure because I told you, like in our family, right? In our family there is different rules because every family is autonomous. I don't have any interfere in your family. So your family is autonomous. My family is autonomous. Likewise, if I am developing the database, there is my own rules and regulations. I can adopt any technology. I can insert as many features as I can. So that is known as all the databases available throughout the world in the scientific community. They are autonomous in nature. Inconsistent in format. Inconsistent in format, yeah, syntax we can say. Why it is known as? Since the biological databases are not unique kind of, right? All the databases are of different types. So obviously, as I told you, the heterogeneity of the data, right? So the data are heterogeneous. So obviously, if you are dealing with that particular data, then different technology you need, the different type of work is. So dear students, due to some urgent work, uh, now I am stopping my session for today. The characteristics of the biological databases, I will see you tomorrow, right? Some urgent work, that's why I have to stop today's lecture. So characteristics of biological databases, I will let you know why. I think most probably the time will be same. At sharp 1 o'clock, I will take your lectures. So and then I will discuss the characteristics of the biological databases. So till then, take care. See you tomorrow.